Hi, good afternoon, everyone. My name is Danielle Edwards. I am the Director of Community Engagement at Jeff Foundation. Thank you so much for joining our webinar series today. We'll be starting up in a few minutes here just to let some of the more attendees join. Okay, I think we've got a bunch of people on now, so we can kind of kick things off again. Again, everyone, thank you so much for joining Jeff Foundation's Community Webinar Series. My name is Danielle Edwards. I'm the Director of Community Engagement. As some of you may already know, our Community Webinar Series are here to help ensure our Duchenne community is better prepared on relevant educational topics. While I wish we could be doing this in person at our family workshops, I'm so glad to be able to provide you with these virtual webinars today. We are so excited to have Brianna Gross today with us, who is a genetic counselor at the Children's Hospital of Philadelphia, who will be presenting on understanding Duchenne and genetic mutations. Please make sure to drop any questions you may have in the chat section below, so Brianna can answer any questions you may have after the presentation. With that being said, I'll toss it over to you, Brianna, and thank you so much again for joining us.
Hello, everyone. Welcome. Thank you so much for being in attendance today. We'll review Duchenne and the genetics behind Duchenne in today's webinar. Bear with me one moment. I'm trying to change the slides. Here we go. So we'll start from the bare bones with a little bit of a genetics lecture, similar to what you may have had in high school or middle school if you did not pursue biology or a genetics field in your adult life. We'll talk a little bit about the DMD gene and where it is and different types of symptoms that people can experience. We'll review something called the reading frame rule, which helps us predict people's symptoms. We'll talk about inheritance patterns, genetic testing, and some therapeutic options and what might be available to you and your family. I did want to start off by going over some definitions that I use interchangeably. So dystrophinopathy is another term that is a bit more inclusive to refer to somebody who has Duchenne or Becker. Dystrophinopathy is just referring to somebody who has muscle disease due to changes in the DMD gene. Phenotype is somebody's clinical picture, the symptoms that they experience, and an exon is a piece of a gene that provides the instructions to create the protein. All genes create proteins, and an exon is just a small little piece of instruction, similar to like a chapter in a book. And a pathogenic variant is just simply another word for a genetic mutation or misspelling that can contribute to symptoms in a person. So we're going to start from the very beginning of genetics. So in all of our cells, we have a nucleus, which is just simply like our command center, telling the cell what to do when. And inside of this nucleus is our genetic information. Our genetic information is stored into chromosomes, which, as you can see here, looks like a little X figurine. And the chromosomes we have 46 of them that come into 23 pairs, and they contain our genes. And our genes are just little instruction manuals to create proteins, which makes us uniquely who we are. We have genes that, contribute to our eye color, the way our heart beats, the way our muscles grow. And the language that genes use is DNA. As you can see in this image on the top right of it, the letters C, T, G, and A are used, and that's the 4 letters of a gene's alphabet. And here's a little visual of the DMD gene. So we have our DMD gene in our chapter book, our recipe, and ultimately we have the DNA alphabet telling the gene or in the body what to make and how to make it. Here's a picture, again, of our chromosomes. And as you can see, our first 22 pairs are just numbered based on size, number 1 being the largest and number 22 being the smallest. And they come in pairs because we receive 1 copy from our mom and 1 copy from our dad. The last pair is our sex chromosomes, which determine if we're male or female. XX is female, so this is an image of a male who has an X and a Y chromosome. So what are genetic mutations? A genetic mutation is a change in the spelling of a gene that ultimately causes the instructions that the gene provides to the body to not be spelled correctly, and it creates a protein that is not recognized by the body, or the protein may not even be made at all. So here is a little example. If you can't already tell, I really like to use analogies because I think it helps make genetics a lot easier to understand. So on the left, we have my secret cake recipe, including our flour, our baking soda, baking powder, sugar, eggs, milk, vanilla, all of the good things. And then on the right, I have my cake recipe. However, I removed the flour, the baking powder, and the baking soda. Ultimately, what happens when I go to try and make the cake following the recipe on the right, I'm not going to get a cake in the end. I get a liquidy substance that probably smells very delicious, but ultimately it's not a cake that's going to rise and that I can cut. And that's similar to a genetic mutation. If we change the spelling, if we change the ingredients, we might not ultimately get the same end product we were hoping for. And this can cause, different body parts to experience different symptoms. So there are a few different types of basic genetic mutations that I want to review. I also like to preface here that I may interchange mutation with pathogenic variant. Again, they mean the exact same thing, just different language to use. We have deletions, which is where a piece of the phrase or the genetic information is deleted. In this example, the dog ran outside was changed to the ran outside. Ultimately, we don't know what the subject of my little sentence is here. And similar to that, the body doesn't understand what it's supposed to make. It doesn't understand the moral of the story. Duplications are where there's a piece of genetic information duplicated. In this instance, the dog ran outside. Could it have been that there was another animal in that sentence? Maybe. And similar to the body processing a duplication, it might not understand what it is supposed to make or what the moral of that story is supposed to be. And substitutions are when a single letter is changed in the genetic information. So instead of the dog ran outside, the sentence was changed to the log ran outside, ultimately completely changing the story. Here is a little bit more of a practical explanation of a genetic change. So here on the left, we have our normal functioning gene, where the DNA in this gene, I made up some letters that are spelling this made up gene. And then underneath it, we have RNA. And RNA you can think of as the Cliff Note version or the summary of DNA. This is what the body uses to ultimately make the protein. So similar to when somebody might be studying for a big test or reviewing an article. They read through it once, get all of the main information, and then they take out the extraneous information that doesn't really impact the, the subject that they were trying to learn more about. And then ultimately the body uses the RNA to create the protein. In the image on the right, we have the letter A, which replaced the letter T on the image on the left in our normal functioning gene. So this would be our mutation. It would be a substitution. And here we can see that ultimately that change caused an abnormal protein to be made or a non-functional protein to be made. Now we're going to talk a little bit more about the DMD gene. So the DMD gene is a very large gene consisting of 79 exons. You can also think of those like little chapters. And the DMD gene is responsible for creating the dystrophin protein, which is a critical piece in keeping our muscle cells healthy. If you were looking at a brick wall, the dystrophin would be similar to the mortar holding the bricks together. If I replace an ingredient in the mortar, the bricks might not be as stable as you would hope them to be. The DMD gene can cause a very large phenotypic spectrum, or in other words, it can cause a varying degree of symptoms, where some people might experience symptoms quite mildly, and we would call that Becker muscular dystrophy, or they might experience some symptoms more severely, where we would classify that as Duchenne muscular dystrophy. We also have intermediate phenotypes where somebody doesn't necessarily fit on the mild end, nor do they necessarily fit on the severe end. You can also see individuals who experience isolated cardiomyopathy and exercise-induced myalgias or muscle pain and muscle cramping. About 60 to 70% of individuals who have Duchenne muscular dystrophy, it's caused by a deletion in the DMD gene, where a chunk of that genetic information is missing. And about 80 to 85% of individuals who have Becker muscular dystrophy have a deletion. And individuals who have Duchenne muscular dystrophy, about 10% of them have a duplication of the genetic information in the DMD gene, and about 5 to 10% of individuals with Becker muscular dystrophy have a duplication. The rest of the individuals who do not fall into the deletion or duplication can have very small deletions or insertions. They might have a missense or substitution change where a single letter may have been spelt wrong. And some individuals have deep intronic mutations. And what that means is that they may have a genetic change that's not detected by regular genetic testing. So how does the genetic mutation predict the severity of symptoms? The tool that we use is a reading frame rule. This predictor tool is not always correct. It is about 91 to 92% accurate, so it does help us in many cases predict the way that somebody might experience the symptoms of dystrophinopathy. The way that we look at it would be the image on the left. As you can see, everything fits together like a perfect puzzle. And ultimately, we're looking to see if with a genetic change, the reading frame is preserved or destroyed. If the change preserves this reading frame where everything still fits together like a puzzle, we would consider this to be an in-frame genetic change, and it would be consistent with Becker muscular dystrophy. And if the reading frame is destroyed where the pieces don't align perfectly in that puzzle, we would consider this to be an out-of-frame genetic change consistent with Duchenne muscular dystrophy. There is some data that has recently come out that suggests that more individuals experience the exception to the rule in the instance of Becker muscular dystrophy. So here is a little bit more of a clear picture of what the reading frame rule looks like. In this instance, we have a deletion of exons 47 to 49. In this case, you can see that exons 46 and 50 align perfectly together. This would be an in-frame deletion, and we would predict that this individual would experience symptoms more like Becker muscular dystrophy, the lesser severe form. In this image, we have a deletion of exons 53 and 54. But with the exons 52 and 55, this deletion causes those two exons to not align. Therefore, we would consider this an out-of-frame deletion, which is consistent with a Duchenne muscular dystrophy phenotype. Next, we'll talk a little bit about inheritance patterns, female carriers, and the reproductive risks for males and females. So the DMD gene is located on the X chromosome, which is one of our sex chromosomes. Females have two X chromosomes and therefore have two DMD genes, where males have one X chromosome and one Y chromosome and only have one DMD gene. Therefore, females can have a mutation on one of the copies of their DMD gene without ever knowing it, because they have a backup copy of the DMD gene that is contributing the dystrophin protein to their body, keeping them asymptomatic. About 2/3 of the time, we find moms of children who have Duchenne or Becker to be carriers, where they have one mutation on their DMD gene and the other copy of their DMD gene is functioning properly. Even if family planning were to be complete, it is still very important that a mother of a child who has Duchenne or Becker receive carrier testing. This is because there are cardiac concerns for female carriers that are important to monitor. About 20% of individuals who are a carrier have nonspecific findings on their echocardiogram, and about 5% of the time it's a bit more serious and they may develop cardiomyopathy requiring treatment. Therefore, the recommendation is that a female receive an echocardiogram every 5 years, beginning at age 25. Overall, for females, if they are determined to be a carrier of Duchenne or Becker, have a 25% chance of having a child who presents with symptoms, and a 25% chance to have a child who is a carrier. And this would be something for every pregnancy, because one pregnancy did have the DMD mutation does not mean that the next one will not, but it also does not mean that the next one definitely will either. Each pregnancy is its own entity with the same risk for each pregnancy. Another way to look at this statistic, I apologize there, they have a 50% chance if a pregnancy were to be a male to be symptomatic and have Duchenne or Becker, and a 50% chance for a female pregnancy to be a carrier as mom is. There are about 1/3 of mothers who have a child with Duchenne or Becker that test negative on carrier testing. However, we do caution that although an individual in this instance would not have to pursue cardiac surveillance, the recurrence risk, if family planning was something that a family was considering, the recurrence risk is not zero. If a mom tests negative and she has a child who has dystrophinopathy, there is about a 10% recurrence risk. This is due to a genetic phenomenon called germline mosaicism. As you can see here in this image, I have a female standing and I have a square around where her ovaries would hypothetically be. So mosaicism is very similar to a mosaic painting where many different colors come together to create an image. In genetics, mosaicism refers to different cell lines that may or may not have different genetics coming together. We do know that in some women who test negative, they have they actually carry the Duchenne or Becker genetic mutation in the DMD gene in their ovaries, although it is not present in their blood. There would be no way to confirm this unless ovaries were biopsied, eggs were biopsied. So the only way to know for certain if a pregnancy did inherit a DMD change that another child, a mom who tested negative, has would be to do prenatal testing while an individual is pregnant. Males with dystrophinopathy have a different recurrence risk. So if you are a male who has Duchenne or Becker, all of your sons will inherit your Y chromosome, and all of your daughters will inherit your X chromosome. And therefore, they're inheriting the DMD change and will be a carrier. There are very strange, unique genetic phenomenons that can happen. So it's not 100%, but it is quite close to 100% that all daughters would be a carrier in that instance. But there is an almost 0% chance that a son would present with Duchenne or Becker muscular dystrophy because they do not inherit their father's X chromosome. They inherit their X chromosome from their mothers. So how does somebody test for dystrophinopathy? I'll review testing for somebody who experiences symptoms or has a family history. And I'll also talk a little bit about family planning testing. So testing of fetuses or testing of embryos. So currently, I know that this page looks very overwhelming. We have a couple different options, and I'll break it down as best as I can. So we have single gene analysis, which is where we're only looking at changes in the DMD gene. Depending on the genetic technology that's used to look at the DMD gene, it may have different limitations. And that's always something that you can talk to your physician or genetic counselor about if there was any questions, if the DMD gene was looked at entirely and thoroughly. This is used often when the symptoms align quite well in thinking dystrophinopathy might be the explanation for the symptoms that somebody's experiencing. Panel testing is used when we have symptoms that may look like Duchenne or Becker, but the symptoms are not as slam dunk. Yes, this is what we were expecting. So this panel test would include more genes than just the DMD gene. However, the genes that are included on panels are all similar in nature in terms of the symptoms that somebody might experience. Often a muscular dystrophy panel might be sent, which looks at other forms of muscular dystrophy. Sometimes a comprehensive test that looks at all different forms of muscle differences can be done that do include the DMD gene. A chromosomal microarray is another test that either your child may have had done or may have been spoken about in different doctor's appointments. A microarray is just a genetic test that looks for extra or missing pieces of chromosome material. So this is not looking at the single gene level, although sometimes it can find those. Therefore, this test can detect deletions or duplications of the DMD gene and will find about 60 to 70% of cases. Whole exome sequencing is a very large test that is performed, and it looks at all 20,000 genes in the body for spelling differences. And often this test is performed when symptoms that a person experienced are not associated with an identifiable genetic cause, or there was negative genetic testing previously. Sometimes individuals might experience more symptoms than just muscle differences, and therefore their doctors ordered this test, and that's how they came to having their diagnosis. I do want to briefly touch upon RNA sequencing because if you're online, if you're doing research, you may come across this. So this is done typically in the diagnostic odyssey if dystrophinopathy is suspected, but no genetic test, no matter the technology that looked at the DMD gene, was able to find a spelling error in the DMD gene. This can be ordered through research. There are a few research labs that do this, but also has recently become more clinically available has been offered by commercial laboratories. So just again to remind you guys, the RNA is the summary that the body will take after reading the DNA. And the RNA is used to make the protein that the gene was supposed to create. And I did want to include muscle biopsy, as some individuals are diagnosed with dystrophinopathy through muscle biopsy, which is where a small piece of muscle, usually from the thigh, is taken and it is stained to see if it has the dystrophin protein or not. People in the labs will look at the muscle under the microscope with a special staining to see if there is dystrophin present or if it is not present. And this is a non-genetic option to confirm a diagnosis. I did want to talk about genetic testing for people who are family planning. So this can be an option if you are already pregnant or if you're considering getting pregnant in the near future. So if you're pregnant, there are two procedures that can be performed to test a baby, amniocentesis and chorionic villus sampling, which is also referred to as CVS. And for somebody who is not yet pregnant, but considering family planning and future children, pre-implantation genetic testing, or PGT, it used to be referred to as PGD, as in dog, for pre-implantation genetic diagnosis. But recently they changed all of the acronyms to be a little bit more specific. To test for the DMD gene. Monogenic disorder means that there is one gene that causes symptoms in a person similar to what DMD is. Therefore, DMD is a monogenic disorder. So the CVS procedure would be where a needle is placed through the belly if the placenta is forward, or a catheter is placed transvaginally if the placenta is towards the back. So everybody is very different, and there's no way to predict, while getting pregnant, where the placenta will lie. The ultrasound technicians will be able to inform a family where the placenta is and what procedure would be best for them. A piece of the placenta is taken before the placenta is fully formed, which is the chorionic villus. This procedure takes place at about 11 to 13 weeks in gestation, and it is able to test for the DMD gene changes that may be present in your family, because the placenta actually comes from the same cell line as the embryo or fetus does too. Therefore, we would see the DMD change in the chorionic villus, or the pre-placenta is another way to refer to it as. And amniocentesis is another procedure that can be done to test a pregnancy. Amniocentesis is typically performed at around 15 weeks gestation, which is where a needle is placed into the belly, avoiding the growing fetus, and a small amount of amniotic fluid is taken. The baby is completely safe with having a couple tablespoons of amniotic fluid removed. And ultimately, the kidney cells, urethral cells, skin cells all contain the genes that we are looking at and can test for the DMD change, and it would be representative of the baby as well. For individuals who are considering family planning and want to test a pregnancy before they're pregnant, they would be able to utilize the PGT. In this case, it's a bit more involved. Even if a person does not experience infertility issues, the in vitro fertilization is required. And this is because they need to control the rate at which the embryos grow. they ultimately, after the in vitro fertilization, will collect all of their eggs and sperm cells and fertilize them in petri dishes. And when the embryos get to a few cells big, they take a piece, so it's like a biopsy. So they take a couple cells from the growing embryo, actually from where the placenta will eventually be. You can tell that on an embryo, which is quite remarkable. And they test it for the DMD change that was in the family. Not all families are eligible for this, and it is something that you would be able to speak with your doctor, your OBGYN, or if you wanted to speak with a laboratory that does this type of test, that would be a great option to learn more to see if you would be eligible for this. Sometimes they do recommend to increase the chances of implantation, because after the embryos are tested and you can select which embryos you would like to implant, the next challenge is to make sure that implantation occurs. So sometimes they'll test the embryos for chromosome differences to make sure that they have the best chance at implanting. Of note, some insurance companies cover this completely, some provide up to a certain amount of money, others completely deny it, which is also another thing to consider if PGT was on your radar. So next I wanted to talk a little bit about therapeutic options, a brief overview. So currently we have 3 exon skipping available treatments where we can skip exons 51, 53, and 45. And we do have gene therapy in the pipeline, which is all very exciting. I do know that exon skipping in gene therapy is a foreign language to many individuals, so I wanted to take the time to review this a little bit more in depth to see if this is something that you and your family may be able to consider now or in the future, depending on if we're able to see more of those exon skipping therapies or have options with gene therapy. So exon skipping therapy is a little counterintuitive, and I'll start from the very beginning. We're going back to the reading frame. The goal of exon skipping is to restore the reading frame. We want all of those pieces to align. So you are essentially making an out of frame deletion into an in frame deletion. Again, I know that sounds counterintuitive, that we are shortening an already shortened gene, but with exon skipping, we are allowing the body to create a shortened protein instead of a non-functional protein. So in this example, I have an out of frame deletion of exons 49 to 50. As you can see in that image, exon 48 and 51 do not align well. In this case, we're going to skip exon 51, which you can see in the image on the right. In this case, exons 48 and 52 align perfectly, allowing the body to read through this deletion. Whereas on the image on the left, where we have that out of frame deletion, the body is not able to read through, and it simply stops creating the protein. So although each therapeutic treatment, so exon skipping of 51, 53, and 45, they all have different specifications for treatment. However that may be, to understand if a male can be considered for exon skipping, there's 2 main things to look into. The first being if the individual has an out of frame deletion. Exon skipping right now is not looking into in-frame deletions, and this is because exon skipping is to restore a reading frame, and an in-frame deletion, the reading frame is restored. And the next thing to consider would be if you were to skip one of the exons that are currently available for treatment, so exons 51, 53, and 45, would that restore your child or your family member, your friend's genetic reading frame of the gene? And I did want to talk a little bit about gene therapy. I know that it's not clinically available yet, but I know it's a hot topic. So gene therapy is a newer technology where a virus is used to give the body back the gene that is not working properly. One issue that was initially seen is that the DMD gene is very large. It's 79 exons long, whereas many other genes range between 2 exons, 8 exons, 12. So as you can see, that is very different than 79. Ultimately, the pharmaceutical companies had to create a mini dystrophin gene where they were able to select the most important critical pieces of the DMD gene to put it together, but they were not able to include all of the pieces. After an individual receives the virus that has this new gene in it, the mini dystrophin gene, the virus infects. I don't really like that term, but that is actually what is happening. The virus infects all of the cells in the body with the new gene. And a common myth is that you can get sick from this in terms of, you know, whatever disease that the virus was carrying. And that's not true. That virus is inoculated. We do not have any genetic information from the virus original form in it to make somebody sick. And another myth is that the new gene, the mini dystrophin gene, can be integrated into somebody's existing DNA, and that's also not true. The new mini dystrophin gene actually will sit alongside of all of the genetic information, and it will use the machinery that the cell uses to read the genes and all of the DNA that already exist in a person's body, but it will not integrate into somebody's genetic information. So with that, thank you so much for listening. I hope that I was able to clarify some questions that you may have had, and I want to open it up for any questions that you guys might have.
Thanks, Brianna, for that presentation. That was great. As Brianna just said, now we'll take some questions from the audience. Please don't hesitate to ask a question. A question you may be thinking is another question another parent could be thinking. So definitely feel free to stream them through.
So I see a question about gene therapy working. At this point in time, the thought is that it should continue working throughout somebody's lifetime. We are only able to give somebody gene therapy once. And this is because after you give them the virus, their body creates antibodies to the virus. And if we were to give it again, the body would attack it and recognize it as foreign. And therefore, it would compromise the amount of cells that then receive the new mini dystrophin gene. There is a question of what we call a watering down effect, which is where as cells divide and continue to grow and replicate, if that new gene will be watered out and more cells without the mini dystrophin gene would be present. But we don't know enough at this point to know that for certain.
Brianna, do you see that next question? Oh.
Yes. So Duchenne and Becker were previously thought of as very separate entities, very black and white. However, as we've learned more, we know that this is more of a spectrum. There is classic Becker, which is where an individual presents similarly to Duchenne, usually at around, they may not have presented with early motor delays, and ultimately ambulation is prolonged in classic Becker to around 15 years of age and into adulthood. Although Becker muscular dystrophy does have a wider phenotypic range than classic Duchenne. Some individuals with Becker experience very mild muscle differences. They might primarily present with cardiac differences over their muscle problems. And they also can experience isolated high CK without muscle differences. I apologize, my asthma. But ultimately, the classification between Becker and Duchenne is based on clinical observation, based on when certain milestones in disease course begin to occur. In Duchenne muscular dystrophy, individuals usually hit up, they may have had their early motor delays, and then they begin to catch up and make gains, and then they hit a plateau phase where they aren't worsening in muscle symptoms, but they're not continuously achieving these new milestones and getting stronger. And then is when they experience their decline beginning around maybe age 7, 8, and then without steroids, experiencing issues with independent ambulation at around age 10 to 12. But with steroid usage, you can prolong that a couple years. Whereas with Becker muscular dystrophy, those hallmark signs and symptoms are pushed back significantly.
Brianna, I know you've touched on this briefly in your presentation, but could you talk a little bit more about carriers and why genetic testing is important for mothers?
Yes. So I know with many of our mothers that I encounter and get to work with, they are very focused on their child, and we all understand that. Many moms will say, I want to focus on getting my care team set with my child. I can't think about me. I'll do this at another time. But it is really important because with observation and having follow up with a cardiologist, you can prevent some significant health issues from happening. And in addition, you would be able to let other family members know if you are a carrier or if you're not, so that they too can take control of their health and pursue medical attention, a cardiologist, if they are at risk for having heart differences too, if they also were a carrier. And also for their own family planning, that might be helpful information to them. So although with thinking about your child's care and how you couldn't care less about your genetics, the primary focus is on your child, which you understand completely. We still think it's important to have a healthy mom that is able to keep up with their child and to make sure that they're taken care of too, because ultimately that will help take care of their child.
Does anyone else have any questions for Brianna? Right, well, we won't keep you too long. And like Brianna said, her email is right there on the screen. If you have any questions regarding your child or your own health, please feel free to reach out to her. We will also send an email out after this webinar too. Thank you for all those who joined us today. This is a great educational opportunity to just learn more about these types of topics, which are very, very important, I think, especially when navigating a Duchenne diagnosis. We're very thankful to have people like Brianna in the healthcare field for us and supportive of these types of meetings. Please feel free to share this educational webinar with any families you may know who would benefit from this. This will be recorded and put up on our website. The more people who can watch this, I think the better. Knowledge is power in this community. So, don't hesitate to, share it with others and reach back out to us if you have any other questions. Brianna, thank you so much for the presentation. It was our pleasure to have you today. And with that being said, we can close down the meeting. If that's okay with you.
Thank you so much.
Thanks, guys. Have a good one, and we hope to see you on another webinar soon.